Put a mint in your mouth and something strange happens. Nothing around you actually becomes colder, yet your tongue can feel as though its temperature just dropped. The same illusion appears in menthol toothpaste, cough drops, and cooling creams. Scientists have known for years that both real cold and menthol activate the same protein in sensory neurons, but one question remains surprisingly difficult to answer. How can a molecule and a drop in temperature trigger the same sensation? Researchers have now captured detailed structures showing how that protein changes as it responds to both cold and menthol. The protein is called TRPM8, an ion channel embedded in the membranes of sensory neurons. Using cryo-electron microscopy together with functional experiments, the team reconstructed multiple stages of its activation and found that cold and menthol take different molecular routes toward the same final gate. The result is something close to a molecular movie of how your nervous system detects coolness. TRPM8 works like a microscopic door through the cell membrane. When the channel is closed, electrically charged ions cannot move freely through its central pore. When TRPM8 opens, ions flow into the sensory neuron and change its electrical state. That activity can then be transmitted through the nervous system and interpreted by the brain as cooling. TRPM8 is considered the primary cold and menthol sensor in humans and is active across temperatures associated with environmental cooling, but calling it a temperature sensor hides the difficult part. A thermometer can detect temperature because the expansion of a liquid or the electrical resistance of a material changes predictably with heat. A protein has no mercury column and no digital sensor. It is a constantly moving structure made from thousands of atoms. Somehow, lowering the temperature changes that structure enough to open a pore. Scientists have been trying to understand exactly where that temperature sensitivity resides. One tempting idea was that TRPM8 might contain a single small region acting as a molecular thermometer. Find that spot, and perhaps cold sensing could be explained by one localized switch. The new study suggests the reality is much more distributed. Researchers found cold-sensitive amino acid residues spread across multiple regions of the channel rather than concentrated in one simple sensor. That means TRPM8 may detect cold through a network of structural changes. Imagine a suspension bridge. Moving one cable slightly might not alter the entire bridge, but changing tension across many connected cables can shift the structure as a whole. TRPM8 appears to behave somewhat like that. Cooling changes interactions across different parts of the protein, and those changes propagate toward the pore. The researchers could actually watch the pore rearrange. Earlier structural studies had already shown that TRPM8 behaves unusually compared with many ion channels. Instead of a relatively fixed pore simply opening like a hinged gate, its pore region undergoes substantial reshaping during activation. Previous work from the same research group described this transformation as a progression from a wide but non-conducting arrangement toward a pore capable of allowing ions through. The new structures extend that picture specifically across different temperatures. As the channel was cooled, researchers captured states along the path toward opening. The pore did not simply snap from off to on. Different structural elements shifted together, creating what the authors describe as dramatic cooling-dependent rearrangements. Then they compared that process with menthol. Menthol does not cool your tissue enough to explain the sensation it produces. Instead, the molecule binds directly to TRPM8. The new experiments indicate that menthol occupies its binding region dynamically and pushes the channel through an allosteric network toward activation. Allosteric simply means that something happening in one part of a protein changes the behavior of another part some distance away. Cold also reaches the same gate, but it does so through an overlapping rather than identical network of structural changes that explains an everyday illusion at the atomic scale. When you eat mint, the menthol molecule is not secretly removing large amounts of heat from your mouth. It is manipulating the same biological machinery your body normally uses to detect actual cooling. The nervous system receives activity from cold-sensitive neurons, and your brain interprets the signal using the system it already has. Menthol has effectively found the molecular shortcut to cold, but the researchers discovered another intriguing region in TRPM8. Their experiments identified what they call a cold spot, a part of the protein especially important for temperature-driven activation. Mutations affecting this region change the channel's response to cooling. The same location can also interact with allyl isothiocyanate, or AITC, a pungent compound better known for producing the sharp sensation associated with mustard and wasabi. That overlap reveals how complicated sensory proteins can be. TRPM8 is not a machine with one button for cold and another for menthol. Different physical and chemical stimuli interact through a connected structure, influencing one another before reaching the same central pore. This helps explain why sensation is not simply a direct reading of the outside world. Your brain never touches an ice cube. It receives electrical signals generated when proteins in sensory cells respond to the ice cube. Chemicals that activate those proteins can therefore reproduce part of the experience without reproducing the original physical condition. The chili pepper can feel hot even though it has not heated your mouth to a dangerous temperature. Menthol creates the cold side version of that trick. Previous work has shown that TRPM8 can adapt or desensitize during prolonged stimulation, and researchers have investigated both activators and inhibitors as potential ways to alter pain signaling. Knowing the precise structural pathways used by cold and menthol could eventually make that drug development more selective. Suppose researchers want to reduce pathological cold pain without eliminating every normal cooling sensation. A drug that simply blocks the entire channel may produce unwanted effects. But if different stimuli use partly distinct molecular networks, future compounds might target specific regions or states of TRPM8. The new study does not demonstrate such a therapy. It provides the structural map that could make more targeted experiments possible. A molecule such as menthol has a shape that can fit into a binding pocket. Light consists of photons that can be absorbed by pigments. Pressure can physically distort a membrane. Temperature is not a single object arriving at a receptor. It changes the motion, energy, and interactions of almost every atom in a protein simultaneously. That makes it difficult to identify which microscopic changes are actually responsible for sensing it. The distributed cold-sensitive network in TRPM8 offers a possible answer. Instead of requiring one perfect thermometer site, temperature could shift the energetic balance of many connected interactions. Once enough of those relationships favor the open configuration, the channel crosses into an active state. The protein itself becomes the thermometer. This also helps explain why evolution can tune temperature sensation. Comparative studies have shown that TRPM8 cold sensitivity has changed during mammalian evolution, allowing species to adapt their sensory systems to different thermal environments. For an animal living in a cold environment, constantly interpreting ordinary temperatures as an urgent cold signal could be disadvantageous. Evolution can modify the receptor rather than redesigning the entire nervous system. The new structural information gives researchers a way to investigate those differences atom by atom. There are still unanswered questions. Cryo-EM provides extraordinarily detailed structural snapshots, but proteins inside living sensory neurons exist in dynamic membranes surrounded by lipids and interacting molecules. TRPM8 also depends on membrane components such as PIP2, and its behavior can change with voltage, chemicals, and cellular conditions. A laboratory structure is therefore a powerful model of the mechanism, not a complete recording of everything happening inside a human nerve ending. Cold and menthol do not appear to activate TRPM8 in exactly the same way. Real cooling causes widespread structural changes that reorganize the pore through a distributed temperature-sensitive network. Menthol binds chemically and pushes the protein along its own overlapping pathway. Eventually, both routes converge on the machinery that opens the ion channel. That convergence is why two completely different events can feel so similar. One can be an actual winter wind lowering the temperature of your skin. But deep inside a sensory neuron, both eventually reach the same microscopic gate. For years, scientists knew the name of that gate. Now they are beginning to see exactly how nature opens it.